right. So I'd like us to welcome Stacey Weiss, who comes from us from University of Puget Sound. And for the sake of time, I will just let her take over. Forward. Yeah. Forward.
This female currently has about a dozen fertilized eggs in her oviduct. I'm trying to get the point. Oh, there we are. About a dozen fertilized eggs in her oviduct undergoing embryonic development. It's the only clutch of eggs she'll produce all year, and there's only about a 30% chance she'll survive the winter to try again next year. So what strategies does she have to maximize the success of this clutch of eggs? Does she act like a bird and tend to her nest, incubating the eggs, warding off predators? Does she feed the hatchlings? No, she doesn't do any of this, right? She finds a sunny spot, digs a hole, lays the eggs, covers over the hole, and then she takes off. The eggs are left to their own devices for about 8 weeks in the soil with no apparent protection from the dangers in the environment. Add to the problem of them that this species, which is the striped plateau lizard, Sceloporus virgatus, their egg-laying behavior is triggered by the onset of the summer monsoon rains, when warm, wet conditions seem to be ripe for fungal growth in the soil, and this potential fungal pathogens can just skyrocket, right? So how do the babies make it out of the nest alive? This stressful situation and the role of microbes in providing an ecological solution to the problem is the focus of my recent research and what I'll be sharing with you today. So as you likely know, parental care is quite rare in squamate reptiles. So it's documented in only 3% of lizards and snakes. And of those that do provide care, it's much more common in species that give live birth than in those that lay eggs. So egg care itself is exceedingly rare, documented in less than 1% of squamate reptiles. But all of those eggs transition from the inside of the mom to that dirt hole in the ground through the cloaca, which is this tissue that you see here. Sorry for the graphic images, but I like them. So the cloaca is the common endpoint for both the reproductive system and the digestive system. So eggs travel through it and feces travel through it. So the cloaca is sure to have microbes. And I would suggest that during this period of extensive contact between the egg and the cloacal tissue, that microbes are likely to transfer, right, from the mom's cloaca to the eggshell. Similarly, in humans and other live birth species, we know that bacteria transition from the birth canal to the baby, right? So you might have heard like C-section babies and vaginally birth babies have different microbiota when they leave the hospital. Okay, so if you're with me.
Sorry.
Oh, that's some kind of bird.
That's a bird, okay.
Yeah. So if you're with me with the assumption that bacteria are likely to transfer from the cloacal tissue to the eggshell during egg laying, it seems to me we might expect evolutionary processes to favor females that transfer a beneficial microbiome, right? A microbiome that might benefit their babies in some way. So our working hypothesis is here that microbes get transferred from the female cloaca to eggshell surfaces during the active oviposition, and that these microbes protect developing embryos from pathogenic fungal infection. Now, fungal infection is one of the leading causes of egg mortality along with egg predation. And we know in other contexts that bacteria can provide antifungal protection for eggs and embryos. This has been found in a variety of taxa, from insects, crustaceans, birds, amphibians. But very few people have suggested the cloaca as the source of those microbes. Instead, a lot of the work that's been done so far is focused on like very specific maternal behaviors or mechanisms to transfer a very specific set of microbes. But we propose that this transition from the cloaca to the eggshells might be very widespread, right? Because all eggs go through cloaca and all cloaca have microbes. So I've been saying we a lot already, and so I want to take a moment before I go into the meat of things to acknowledge my collaborators and my funding. So I am faculty at the University of Puget Sound. This is a small liberal arts college in Tacoma, Washington, that's focused on undergraduate education and providing hands-on, authentic research experiences for undergraduates. Murdoch Charitable Trust and National Science Foundation has funded my research, and most of my research has been at the Southwestern Research Station in the Chiricahuas, which is owned and operated and supported by the American Museum of Natural History. Three main collaborators have worked with me on this work. So Mark Martin is a microbial biologist at the University of Puget Sound. Betsy Arnold here at the University of Arizona trained me on next generation sequencing and bioinformatic work along with me. Thank you. And Marie Bunker has been my technician for 6 years. Sadly, funding ran out 2 weeks ago. So if anyone's looking for a great scientist to hire, looking for a job. And then I worked with a whole slew of wonderful undergraduate students, as well as some high school students, Southern Arizona high school students, thanks to my relationship with Margaret Welch. Margaret was a teacher at Tucson High Magnet School.
My science teacher.
Oh, really? Yeah. Oh, Margaret's so awesome. And then when she left Tucson High, she went to SARSF, which is a regional organization that works to provide authentic research to high school students. Okay, so what have we done? Oh, first, our study species. Study species, the striped plateau lizard, Sceloporus virgatus. This species is found mostly in northern Mexico, and it gets into the United States just in these borderlands between Arizona and New Mexico and Mexico, specifically in these three mountain ranges, so the Chiricahuas, Palancios, and Animas mountains. And I am very fortunate to have studied them for a long time, longer than I'm willing to admit, up this beautiful canyon, Cave Creek Canyon in the Chiricahuas. And we stay at the Southwestern Research Station, which hopefully you all have been to before, or at least many of you have enjoyed what the research station has to offer. I'm trained as a behavioral ecologist, and most of my work on the striped plateau lizard has focused on the fact that females have this bright orange ornament that they develop along with the ovarian cycle. So most of my work, until I started this microbial stuff, was looking at the function and regulation of this female ornament in the context of sexual selection. And while I did this work, every now and then we would convince females to lay their eggs for us. And in the lab, we would have really good hatch success of these eggs. But every now and then I would notice some fungus beginning to grow on the eggs. And when I saw egg mortality was always associated with a heavy fungal infection. And anecdotally, I noticed that fungal growth was 4 times more likely to occur on eggs that I had dissected out of the female for whatever reason than those that had been laid. And I mentioned this in casual conversation with my microbiologist colleague, Mark Martin, and he said, "Coical microbes." And that basically started all the work I'm going to tell you about today. So follow interesting things in your work. Okay, so we've taken a multi-pronged approach to addressing our hypothesis, and I'll kind of walk you through our key findings for each of them, starting with a more direct comparison between laid and dissected eggs, right? So these are eggs that go through the cloaca versus eggs that don't go through the cloaca. So to do this, we collect females, we swab their cloaca, we swab their eggs, and those swabs are gathering up the microbial community, right? And then we do a number of different things with those microbes. The main technique that we've used and what Betsy and Ming has helped me learn is next generation sequencing on the Illumina platform. So what this means is we take our sample, we extract the DNA, we amplify the DNA through PCR, barcode it, send it off to a lab for Illumina sequencing. They send us back millions and millions of short DNA sequences that we use bioinformatics to go through quality control and to identify each of those little sequences to a bacterial taxonomic group. And then we can analyze things like alpha and beta diversity of the microbiome. In this study, we also did some plate-based work. So we, swabbed the sample onto plates and saw what grew. And then we are fortunate at the University of Puget Sound, a relatively small school. We have a scanning electron microscope that our undergraduate students can get trained on and actually operate themselves. And so we use the scanning electron microscope to actually visualize the bacteria and fungus on the eggshells. Which is pretty cool. Yeah. Then we took the remaining eggs and incubated them to hatching. We have cute little babies, and we measure whatever we can on those offspring. So here's our first data slide. The y-axis here is eggshell richness. So what this means, it's essentially the number of types of bacteria that we find on the eggshell. So it's kind of like the number of species, but species concepts and bacteria are weird, but it's like the number of species that we find there. And just ignore the x-axis for now. Just focus on the y-axis. You can see that the orange dots, which represent the laid eggs, have more richness. They have a richer microbial community than do the dissected eggs and the blue dots. Okay, so laid eggs get more bacteria, or at least more types of bacteria. And now if you pay attention to the x-axis, this is the cloacal richness of the mom. And you can see this positive relationship between the mom's cloacal bacterial richness and her eggshell bacterial richness, suggesting that those bacteria are actually coming from the mom. That's how we interpret that. And that relationship is totally lost, right, in the dissected eggs. So we think this is good evidence that bacteria are indeed getting transferred from the cloacal tissue to the eggshells. Next, we can go to the SEM and we get beautiful pictures like these. So we can see little tiny bacteria, fungal spores, and fungal hyphae on the eggshell surfaces using the scanning electron microscope. I especially like this image here because if I zoom in on that one region, hopefully you can see there are bacteria kind of associated with this fungal hyphae. And you can almost see little like pockmarks, like maybe those bacteria are working to break down the fungus. Intriguing. Okay, so this is work from student Grace Elliott. And before I show you her results, I just want to check in. This is your test. Make sure you're all with me. So are we predicting to find more bacteria on the laid or dissected eggs?
Laid.
The laid ones, right? Because they go through the cloaca. And how about the fungus? Are more fungus going to be on the laid or dissected eggs? Dissected, right? Because they don't get the benefit of the mom's antifungal microbes. Okay, great. All right. But this is what we find. So here we see the amount of bacteria on the eggs on day 0, which is the day we acquire the eggs, right? Either through oviposition or dissection. More bacteria on laid versus dissected. Then we incubate the eggs in an environment that includes soil from their natural environment. And 25 days later, we dig them back up and we find more fungus on the dissected eggs and the laid eggs. And then Grace went a step further. She took just little tiny pieces of the eggshell and incubated it directly with a solution that had fungal spores for 48 hours, then rinsed them off and looked at those under the scanning electron microscope. And with 2 different types of fungus that we isolated from previous failed eggs, Neocosmospora and Aspergillus, we find more fungal attachment to the dissected eggs than the laid eggs. So pretty cool. I was excited about that. All right, but is it going to actually affect hatch success? Like, does it actually matter? I don't know yet. Well, let's find out. So we turn to Helena Higher-Gray's work. And here you can see dissected eggs definitely have reduced hatching success. And the impact of dissection was more severe the more realistic the incubation environment. So in the white bars, we have a sterile environment. The gray bars, we start with the sterile environment and we added back in some fungal spores, just kind of made a little cocktail. But with the black bars, this is where we added in the soil. So whatever the natural complement is kind of went in with the eggs. And you can see a really strong reduction in hatch success. What was also cool is that Helena found that those hatchlings from the laid eggs were also longer, bigger, and heavier than those from dissected eggs. And what this indicates to me is that those embryos developing in the dissected eggs couldn't allocate as much energy to growth. Maybe they were allocating energy to like immune system activation. I don't know that, but that would be cool for someone to test directly. Okay, so far, we know that relative to eggs without coequal exposure, our laid eggs have a microbial diversity more like their moms, more bacteria, less fungus, and higher success. Okay, but is that higher success actually due to the bacteria, or do we just have two things that are kind of happening in parallel, but they're not causal? This is our next step. So to address this question, we did direct inhibition assays. So we take bacteria that we isolated from the cloaca or directly from eggshells. We take fungus that we isolated from soil, and we pit them against each other in a weird little fight club in the lab. We started out with this word really qualitatively, and as an ecologist, qualitative things aren't my favorite, but this is where we started. So this is actually work from Tucson High students. So here on this plate, on the right side of the plate, this isn't a one strain of bacteria, but they just took a swab of the mom's cloaca and spread it all across the plate, and whatever bacteria would grow, grew. And then they placed a little bit of fungus right here and looked at fungal growth. What you can see is the fungus is clearly only growing away from the bacteria, and we have these zones of clearing on either side. What this indicates to us is that the bacteria are secreting something, right, that is preventing fungal growth or inhibiting fungal growth. So that was really cool. Yeah. But we wanted to go, kind of do a bacterial strain by bacterial strain and try to get things kind of ramped up in that way. So these are two of my undergraduate students, Olivia Prodi and Helena again. And what they did is they took a plate, they grew a single species of fungus kind of all over the background. And then they patched on like different strains of bacteria. And again, we're looking for that zone of clearing. So you can see on these two plates, bacteria 3 led to a zone of clearing. Cool, but I wanted to get quantitative. And so this is where my student, McKenna Boulay, stepped in. And so McKenna was able to get us more quantitative in two ways. One is that she quantified like the amount of bacteria and the amount of fungus that we put on a plate. So that was step one. And so in these plate-based assays, we have a known amount of bacteria and 1,000 fungal spores in the center, and we see if the fungus can grow or not. Step forward, but not as quantitative as I want to be. Here we put in a known amount of bacteria with a known amount of fungal spores. We incubate it for a set amount of time. And now we can actually get quantitative. We can take a sample after the incubation period and actually look at it under the microscope, using an instrument called a hemocytometer, and actually count the amount of fungus that's there. So that was exciting. And McKenna is no slacker. She did this with 7 bacterial strains, 4 fungal strains, so all 28 combinations. Each combination has 5 to 10, sample size of 5 to 10, with both methods. So a lot of work. Yeah. McKenna's awesome. Yeah. Okay, so here's her data for our 7 strains of bacteria that we're interested in versus Fusarium. So I'm showing you Fusarium because it is one of the most important pathogenic fungi worldwide. It impacts plants, animals, and it's also a known killer of sea turtle eggs. So we know it has direct mortality in reptiles. Okay, so these are her plates. This is just a control, nothing's on it. This is a control, just fungus, no bacteria. So if the fungus isn't inhibited by the bacteria on our other plates, it should always look like this. And then we have 1, 2, 3, 4, 5, 6, 7 other plates with a different type of bacteria in the background. Does the fungus grow? You can see it only grows here on the 3rd bacteria and the 7th bacteria. And now if we look at the liquid co-culture data, these are actual spore counts, we see that 3rd bacteria and the 7th bacteria grew pretty similar to the control here on the far left, but those other 5 species of bacteria significantly cut down the fungal growth. Great. The other, the bacteria versus the other 3 bacteria look pretty similar. I just have it here in a table. Our 4 fungus, our 7 bacteria. These were the only 4 interactions that were not significantly, where we did not see significant inhibition of the fungus. All the others, we see significant inhibition, and those cells shaded red have at least 95% inhibition of the fungus. So it's a lot. They did a really good job. Okay, so we can add into our summary that indirect challenges, the colloidal and eggshell bacteria certainly inhibit fungal growth. Okay. New directions that we're taking this work, we're interested in looking at the mechanisms that the bacteria are using to prevent fungal growth. And one of the first things that we were looking at is the role of chitinases. So chitin is one of the main components of fungal cell walls. Chitinases would be enzymes that break down the chitin, right? And so other types of bacteria are known to secrete chitinases. So we thought this might be the situation. So this is Temple D. She's a Southern Arizona high school student that I work with through SARSA. And she grew the same 7 strains that McKenna was using on a plate where the auger here, we called it chitin minimum media. The only nutrient source on the plate was chitin. So the only bacteria that can grow, presumably, right, are those that can break down chitin. And you can see that at least 3, maybe 4 of these bacteria were able to grow on this plate. Since Temple's work, we've taken 1 species of bacteria and done whole genome sequencing, and we do indeed find the gene for chitinase. So I'm also interested in kind of stepping away from the antifungal benefits of the host microbiome to think about other ways the lizard host might benefit from its microbiome. And you might know in the literature, there's all sorts of examples of this from, everything from mental health to disease, right? And so one of the first things that we're doing there is thinking about interactions between the host microbiome and the host immune system. And so this is work that I'm doing with Susanna French from Utah State University. We're just kind of getting started on it. Okay, so if our hypothesis is true, I have a couple of other things that I would expect to see in terms of the microbial profile. We haven't really talked about like what types of bacteria are there or not there. So if my hypothesis is true, I would expect that the cloacal microbiome would be highly specialized relative to other tissues higher up. So relative to like higher up in the intestine or higher up in the oviduct, right? The microbiome should be special in some way, right? If it has this very specific evolved function for egg protection. So we might expect the cloaca to be special. I might expect that the female cloacal microbiome is special, right? Because I wouldn't necessarily expect these antifungal microbes in the males. They're not laying eggs. And I might even expect that the female cloacal microbiome might be most special right around egg laying, right? That she might not have these antifungal microbes all the time, but as she prepares for egg laying, we would see a shift in the microbial in some way. Okay, so let's dive into what we know about these things. First study was to compare the cloacal and oviduct, the cloacal tissue to oviductal tissue and intestinal tissue. So we have cloaca, lower intestine, upper intestine, oviduct. What's being plotted here is the mean relative abundance of bacteria that were identified as being members of different phyla. So when we're talking about phyla, this is like comparing vertebrates to worms, right? So that's what we're talking about in terms of taxonomic level when we're thinking about phyla. And so you see there are 3 main phyla that are relevant to us: Proteobacteria, Firmicutes, and Bacteroidetes. And Firmicutes and Bacteroidetes are commonly considered to comprise the core of the vertebrate gut microbiome. So if you read about like the human gut microbiome, you'll see these terms come up a lot. But most of that work has been done in humans and mammals. As more work is being done on wild microbiomes of non-mammalians, we're finding that proteobacteria is actually much more common in the guts of those taxa relative to mammals, including humans. And in those species, so there's a couple of predatory birds, a snake, and now Sceloporus virgatus. We're seeing the amount of proteobacteria increases as you move down the upper intestine. So as you move down the intestinal tract, from the upper intestine to the lower intestine to the cloaca. So that's also been found in other species. But this amount of specialization is insane, right? So 99% of the bacteria that we isolate from the cloaca were from this one phylum. I haven't seen that in anywhere else in the literature. So we definitely seem to have some important specialization going As an aside, we also looked at the microbial community in the feces, and you can see that the feces don't estimate any other tissue within the animal. And I point this out because there are many, many studies being done where people say they are studying the gut microbiome, and what they're actually studying is the fecal microbiome, and they have not done the work to validate that. I'm not saying that in no case might the feces actually give you an indication of what's going on in the gut, but it's worth documenting it, and most people don't. In this case, certainly if I was just saying the fecal microbiome, that would be telling me a totally different story. Our main approach to getting our samples is by colloidal swabs, and so here are some examples of colloidal swab results. And you can see they're not statistically different from the cloaca, but they were more similar to the lower intestine. And we think in this study, we actually had some fecal contamination on our swabs, which is a common problem with swabs where you stick something up.
Yeah, right.
Yeah, it's not uncommon that they like are upset with you and have some poop action going. But since we've published this study, we've come up with a bioinformatic approach to try to remove fecal contamination from our samples. And so we're hoping that we don't have that fingerprint in our few work since this publication. But clearly, there was no significant difference at least. So our colloidal swab samples are certainly doing better than our fecal samples. Okay, we're going to move down to the family level of taxonomic classification. And so with families, like this is comparing rhinosomatids to teiids, right? So spiny lizards to the whiptails. And the data here, you know, there's a lot of colors, whatever. The main thing I want you to, hopefully you can see that like and here I have every sample, it gets its own bar, whereas the previous graph, I just showed you the averages. But hopefully you can see just kind of big picture, the oviduct is different than the upper intestine, and the upper intestine is different than the lower intestine. The lower intestine looks pretty similar to the cloacal swabs, and then here's the cloacal tissue with extreme specialization. So the dominant family in the maternal cloaca is Enterobacteriaceae, with 83% of the bacteria belonging to a single family. This is like taking the Sonoran Desert, all the animals, maybe even just all the vertebrates we can find, and they're all rhinosomatids. 83% of them are rhinosomatids, right? That's crazy, right? It's interesting, Enterobacteriaceae is known to be, to often be symbiotic in intestines of other species. They have, some of their members are known to have antifungal properties, as well as other benefits to the host. So it's perhaps not surprising that it's Enterobacteriaceae that's showing up here. And of those 7 strains that McKenna was using on the previous slide, the bacteria that have the strongest antifungal action are also in this family, Enterobacteriaceae. So yeah, I think the cloacal microbiome is special in some ways, certainly different from other areas of the reproductive and intestinal tract. Okay, so it's the female cloacal microbiome special. Is it different from the males? Here we're comparing, it's about 130 males to about 130 females. And no difference at the level of phylum, and not a significant difference at the level of family, but at least there's like a little bit more of the Enterobacteriaceae in the females than in the males. So we have to go to a lower level, or a more detailed level of taxonomic classification, to what we call the ASV. So ASV stands for amplified sequence variant. It's like the closest we can get to species, or maybe even subspecies. And what you can see with this simple Venn diagram is we have about, close to like 1,900 different ASVs, or types of bacteria. You can see males and females only share about 20%. So they are different at this fine scale level. And this plot is a lot to take in, but what this plot is doing is it's taking like the 1,900, the variation in these 1,900 different ASVs, or types of bacteria, and kind of squishing it down to 2 axes. So each of these dots is a different microbial community from a different host lizard. And the blue dots are females, the red dots are males, and it's like this big fireworks display. But here, these white dots are showing the centroids, and this is statistically significantly different. It's not as beautiful as I would like it to be. Like sometimes you see these plots and it's like males, females. I'm like, oh, the diagram of my dream. So I don't have that, but I do have statistical significance. So I'm running with it and saying that yes, the female colloidal microbiome is special relative to males. But again, at this like finite level of classification. Okay, so does the female colloidal microbiome shift with the ovarian cycle, and does it get more specialized during as she prepares for egg laying? So a quick review of the annual cycle of the female striped plateau lizard. So right now they are in winter dormancy. Females should emerge around April, start eating, setting up territories, and this is the pre-reproductive stage. In May, they begin follicular growth, so the follicles start growing on the ovary. We call this the vitellogenic period because vitellogenin is this protein that's important for yolk production, and so it gets called vitellogenic. After females ovulate, the eggs burst out of the follicle, go into the oviduct, where they stay for about a month. And while the eggs are in the oviduct, the embryos begin developing, and we call this the gravid period. And then the rains start, the females will oviposit within a week or so of each other, and then we have this post-reproductive period where they're eating and getting ready for winter dormancy again. So what we're going to do is we're going to compare the colloidal microbial community of these 4 different groups: pre-reproductive females, vitellogenic females, gravid, and post-reproductive. There is no difference at the phylum level, so we'll just start here with the family level. Not statistically different again, but I'm intrigued by the fact that Enterobacteriaceae, this key family that we were talking about, does increase from pre-vitellogenic to vitellogenic to gravid, and then decreases again after egg laying. So suggesting maybe we didn't have statistical significance. So when we look at these crazy fireworks displays and we focus on the centroids at these 3 different reproductive states, we can see the females transitioning from pre-reproductive to vitellogenic, and then they kind of shoot up here when they're gravid and then fall back down when they're post-reproductive. And this was statistically significant, showing that there is this shift during the gravid stage. So I'm saying yes. Okay, so this is all supportive of our hypothesis that our next steps are trying to think about how this specialized microbiome gets acquired, how it gets regulated, like what's controlling the shift from villogenesis to the gravid state. We don't know. We did start by looking at the role of vertical transmission, looking at all the maternal tissues versus the offspring tissues. I purposely kept this small, so I won't tell you about it, other than saying that we've done it. But if you have questions about it, I could show you later. Our last approach. So again, if my hypothesis is true, I would expect these specialized microbiomes to be present in egg-laying species, but not in live-bearing species. Right? Live-bearing species, as far as I can think through, like don't need antifungal microbes to transfer to their babies. So Sceloporus virgatus is found sympatrically with Sceloporus yearvi, and yearvi happens to give live birth. And so my goal was for the summer of 2020 was to get down to the study site and sample some yearvi cloaca. But before I could get to Sceloporus yearvi, COVID came to all of us, and I was stuck in Washington for a summer. Oh my gosh, that was hard for me. I hadn't had a summer without Arizona for a long time. But being pros at turning lemons into lemonade, we put on our masks, we hiked around with masks on, and we looked for our local Washington species of Sceloporus occidentalis, which we did find in a couple of different places. And this expanded our comparative approach. And so now we can also consider the role of host genetics and the environment. Because we have this 2 species comparison I already told you about, right? Virgatus and yearvi, egg laying versus live birth. They're sympatric, they're found in the same place. They're both Sceloporus, but they're relatively distantly related. So within the clade of Sceloporus, they're separated by like 19 million years. In contrast, Virgatus and occidentalis, they're both egg laying. They're actually sister species on the phylogeny, so they're most closely related to each other, which was convenient for me, but they're allopathic, right? They're in very different places. So we have this 3 species comparison, but we were in Washington all summer, so we didn't get one population of occidentalis, we got 3. So we have a beach site where the lizards were running up and down the driftwood, and I got to see whales when I was out doing field work, which like I never thought that would happen in my life. And then we had a forest population, which was like a half mile away from the beach, just kind of up the hill. And then about 150 miles inland, Bear Canyon, which reminded me a little bit of Cave Creek while I was there and missing Cave Creek Canyon. And so I consider it kind of the same-ish habitat as Cave Creek. So I'm calling this one habitat type. I know it's a little bit of a stretch, but bear with me. So I have my 3 species comparison with one habitat type, if you'll give me that. And then one species, occidentalis, with 3 different habitats. All right, so I'm just going to show you the data in this tree form. So this is a dendrogram, and what it's showing is summarizing the microbial community for each of our populations and looking at how similar they are to each other and like giving this tree-like structure. And what you can see is the virgatus is clustering with Yerovide, the one that it lives with, but is very different from. And then we have our 3 occidentalis populations down here. So based on this, it seems like the shared environment may be more impactful than shared reproductive mode or shared evolutionary history. But if we just focus on that phylum proteobacteria that we talked about at the start, it was more common in the egg-laying species than in the live-bearing species. And so I feel like there's something more there to investigate. Still a lot to learn. And so some of the new directions that I'm taking here is to expand this comparative approach to include more species, right? This is just 3 species, not a whole lot. So I joined a collaborative team led by Amelia Martins at Arizona State University, includes Michelle Lang at Texas A&M and Jaime Zuniga-Vega at UNAM in Mexico City, as well as a slew of technicians, postdocs, grad students, undergraduate students. And we conducted fieldwork in Texas and in Oaxaca. And I now have samples from 11 species of Sceloporus. And so with these species, I have a number of different goals. So one is really to dive into this role of reproductive mode. I still feel like something might be there, comparing oviparous versus viviparous species. And then we're also interested in using this data set to look for something called phylosymbiosis. Phylosymbiosis is this idea that has emerged from all the new microbial ecology work that suggests that if the host microbiome is kind of co-evolving with the host, that if we look at the the phylogenetic tree of the host, so here it's flies, but imagine these are different species of Sceloporus, and then we take the microbial dendrogram, like I showed on the last slide, of their microbial communities, phylosymbiosis would be this idea that the tree structures parallel each other. Clearly, we did not see this in my 3 species study, but I'm excited to see what we find when we have 11 species, right? Maybe this pattern might emerge. And then we're also interested in looking at the role of the shared environment. And in here, when we were in Oaxaca, we found Grammicus, Sceloporus grammicus, and spinosus. So we went to 80 different sites. In some sites it was only Grammicus, some sites it was only spinosus, and some sites they were together. And so we want to see if the microbiota of the 2 species are more similar when they're living together than when they're apart. And tomorrow I head up to Arizona State to analyze data. So maybe I'll have some answers real soon. Okay, so through this multi-pronged approach, I think we have a variety of lines of evidence to support our hypothesis that microbes are being transferred from the cloaca to the eggshells and that they're serving to protect the embryos from pathogenic fungal infection. And we propose that this might be a quite common source of egg protection. Again, as I said, right, all eggs go through the cloaca and all cloaca have microbes, but it might be especially important in those species that lay and leave and have no apparent parental care. And the other thing to consider is that maybe we need to expand our conceptualization of what parental care is from including, oh, sorry, I also have lots of different new directions. To broaden our conceptualization of what parental care is from all of these wonderful behaviors to also including this simple behavior, right? Maybe this is representing parental care. Thank you so much, and I look forward to questions.
Yeah, so the Sceloporus species you've worked with, how has climate change affected their reproductive behavior, offspring survival, and bacterial composition?
Yeah, it's great. I don't know. I don't know. I think there's some climate estimates that suggest that Sceloporus as a genus is in danger from climate change. I know my populations in the Chiricahuas were really low last year. I don't know, if it's climate change or not, but the populations are declining. Reproduction seems to be reproductive success seems to be dependent on winter rains. And so like a really dry year like this, I'm expecting really low success, this coming season, which is a bummer because my sites were already low. How climate change affects the microbiome is a really interesting question. There are a few studies starting to come out and showing some effects, but for this specific example, yeah, I don't know yet. But we're interested in looking at, virgatus doesn't have a huge elevational gradient, right? They're kind of really at this kind of 5,500 foot elevation. We did take a sample of Yarraby from as low as we could find them up to the top of Rustler, and we're interested in looking at an elevational gradient to try to get at the role of temperature and climate. But yeah, it's an interesting question. I don't have the answers yet.
So is there a difference in bacterial composition with elevational gradient?
We haven't analyzed the data yet, so stay tuned.
Looking forward.
Yeah.
You're saying that the soil is a neutral factor. Is there something that maybe the animal selects a certain type of soil instinctually?
Yeah.
That plays a role in
Yeah.
The load of fungal load.
It's a great question.
Egg is subjected to.
So the females, we did some radio tracking because it's really hard to observe their nesting behavior. So we tracked a whole bunch of lizards, a ton of people hours to get 4 nests. So it's a lot of work. It's hard to get that information. But we do know, so females are, their territories are along the creek bed. And my study site, it's the creek and the plateaus and then these upper slopes. And then there's the high plateau, and the lizards are not really on the high plateau at all. They kind of stay within the boundaries of the creek bed. But to lay their eggs, they run up those hills up to the higher plateaus and they lay their eggs on the higher plateau. And I think that's because the soil there might be drier and it might avoid catastrophes from flash floods, right? So it might be this evolved behavior to go up out of the creek bed. We do find that females occasionally will start to dig and then they'll stop and run to a different spot and dig again. What it is that makes them change their mind about the location, I don't know. If they could be detecting something about the microbiota of the site or just the moisture of the site. There is one female, we had a couple, I think 2 of our females dug and then moved and ended up laying somewhere else. And at one of those 2 where the female abandoned a dig site, when we dug it up, there was termites everywhere. So that was probably why she left. But the other one, I don't know. Yeah, but they're definitely they're moving about 30 meters away from their home range to their nest site. So they're going somewhere, they're looking for something else. Yeah.
So I think you found some difference in the microbiome cloaca females versus males.
Yeah.
Wasn't huge difference.
No.
Did you look at the seasonal shifts in the males? See if they mirrored the females?
Yes, I did. Sorry for what we found. We did look at it because I did this study during my sabbatical when I was here at U of A for the year. I was going to the research station every 2 weeks. So I have the whole active period of the lizards. I'll look at my paper and I'll get back to you on that.
I think a related question.
Yeah.
That's a seasonal shift.
Yeah.
Do you have a sense of whether that's because of internal hormonal changes or because of exterior environment changes?
Yeah, we did not sample the soil, throughout that period. I don't think their diet is shifting a lot, over the active season. So my instinct is that it's more like, hormonal regulation that's causing the shift, but I don't know, and I would love to find out.
So I'm curious about the hatchlings, right? I'm assuming once you guys had the hatchlings, did you guys ever swap the hatchlings cloaca too to see if how much of that microbiome also transferred over?
We didn't collect enough, material from them. They're so small, like we can't get a swab in their cloaca, they're like too small for that. And so, well, I guess that's not true. So in our maternal transmission study, we actually did find this by sacrificing some of the hatchlings and looking at like the full intestinal tissue. And so, the microbes that we found in the hatchling intestines seem to be sourced from both the maternal cloaca and the maternal oviduct. So some of those microbes might be getting transferred, during the gravid period when the eggs are still inside the oviduct. There might be some microbe transfer at that point, but then also at the egg line stage.
Have you compared the microscopic texture of the eggs across species?
Not across species, but not touching anything. Let me see if they're here. I have them.
I wonder if that has anything to do with being able to capture.
Oh, yeah, sure.
Yeah.
Possibly, yeah. So I mean, these are some images of eggshell surfaces. Like, I would love someone who knows this stuff to look at them and tell me what we're seeing. But we see like, some like kind of crust and trough morphology that may be, places that can catch, kind of catch the bacteria. And then my student found these weird little spheres that disappeared over incubation. So they were more common like when the eggs were young than when the eggs were ready to hatch.
Yeah.
But I don't know. I look at the images, I'm like, I don't really know what's there, but I need an expert to come and analyze them for me.
And yeah, and Dale kind of alluded to these changes, like the relationship between like hormonal changes and the tissue and the relationship between the tissue and the microbes.
Yeah.
Is there something? What's the relationship between the bacteria and the tissue and any hormones or physiological things that are happening?
Yeah, and I'm sure the environment is really different. Like, I mean, the pH might be different at the cloaca versus the lower intestine, or the acidity, right? So there could be things that help kind of winnow the microbial community, right?
Yeah.
So that it's really specialized once you get to the cloaca.
I can imagine climate change and drought have significant impacts.
Yeah, that seems likely.
Following up on the question back here, on the transfer of those antifungal microbes that go into the hatchling, is that because the egg would crack and it would be on the hatchling itself? And if we're looking at viviparous species, are those truly viviparous or are they oval?
Oh, yeah.
Have an egg inside of the cloaca
Right.
and not just the placenta.
I think Europe might be ovoviviparous. Yeah, so they're certainly hanging out in the mom's oviduct for a while.
Yeah.
Right. So, I mean, the maternal oviductal community and the cloacal community could be affecting the baby's microbial community, but I just don't expect it to have an antifungal protection function. But I imagine there's still vertical transmission of the mom's microbiome to the hatch. But yeah, is that what you're saying?
I'm just wondering, are you aware of any speculative viviparous species where you'll have a viviparous
Oh, yeah.
population and
I think they're. I don't know what's.
Yeah, but if there's
Oh, no.
Not, yeah.
I don't remember if I've heard an allusion to that happening in
Yeah.
Fleveni, but I don't
Oh, it's not nice.
I
I don't
don't
know,
know.
but that's the dream. That would be the dream. Yeah, that'd be really cool. I don't know if there's, maybe not a phrynosomatid, but in a different taxonomic. I feel like I've heard about this.
asking people.
Okay, yeah, that's Google. Ask Siri. Yeah, that'd be really cool. Let me see, there's a Zoom question.
Those are the only two Zoom questions so far.
Okay, what would be the physiological mechanism that increases beneficial bacteria based on season? Yeah, so that we've already kind of talked about a little bit. I don't know, but I think maybe, hormonal changes going on, during the ovarian cycle would be an obvious place to look, just since we have the cycling of the bacteria along with the cycling of the female reproductive state. And did I miss error bars on the graph on the slide or the first summary slide?
I think there's one more question before that one too.
Oh, okay. I'll scroll up. Let me look at this one. Slide. Oh, here. Yeah, there's no error bars here because this is percent hatch success. So it's like the number of hatchlings of all the eggs, the percent that hatched. So I don't have error bars there. And do you consider the production of antifungal bacteria as commercial investment to incur a cost to the parent? Or defined for investment, or is it free? Yeah, so I mean, it's a good question. I mean, I'm proposing that maybe we could consider it part of parental care, but whether it's costly, I don't know. I mean, there's a clear fitness benefit to it, so I don't know how the costs and benefits would weigh out. I'll have to ponder that one, but I mean, I guess my working hypothesis is that it's a net benefit to the females, right? Otherwise, the specialized microbiome.
Apparently, the Australian three-toed skink is a species that can both lay eggs and give birth to livestock.
Anyone know someone in Australia?
It's called bimodal reproduction.
I did also find it wasn't Slevini, it was another member of the Sceloporus complex, but
Oh,
Sceloporus bicanthalus
oh.
apparently has both ovoviviparous and viviparous population.
See, I wanted to catch you while this chart is up.
Yeah.
What kind of hatch time are you talking about with these? Do you get any kind of variance between, those you surgically remove and?
Oh, yeah. We analyzed hatch time and there was no significant difference. And in this case, obviously the hatch time depends on the incubation temperature that we use, but in this case, it was like 42 days, something like that. Yeah. No, because I was really interested in that to see if one developed faster than the other.
So how many species do you have of Sceloporus of live specimens at your lab?
At my lab, I have 2 occidentalis. Yeah, I don't have a lot in the lab. Most of my work is all field-based. And then the only reason I have these 2 Sceloporus is that Washington Fish and Wildlife, unlike Arizona Game and Fish, if I catch a lizard and bring it into the lab, even if it's for like a second, I cannot release it. So it's really limited the amount that I've done with occidentalis, because here in Arizona, like I can collect the lizards, I can bring them to the research station for a week or two, do some experiments and release them. And in Washington, I can't do that. And so I don't want to take a whole bunch of lizards out of the wild forever. And so I did it, once, and these lizards are they've been in the lab for 2.5 years, just like hanging out. So
It started to cross my mind because you said there was a species whose population was declining. If maybe a captive breeding program could be set up for population augmentation.
oh yeah, be really interesting. I don't know if anyone here has reared lizards. It's hard for me to keep those hatchlings alive. I've tried everything and it's my success is really low. So it would be a big effort, and I don't think, at least virgatus isn't at the point where I think those efforts are really needed quite yet. But I did meet someone at the Biology of Lizards conference who's successfully rearing some Sceloporus, and I wanted to hear more about his methods because I really struggle with that.
Personally, I've been of the belief that every species and subspecies, when and where possible, of wild animal and plant needs a captive for
Yeah.
sure that's possible, yeah.
Well,
Yeah.
it brings up that, like, this has implications for reintroduction efforts in general,
Yes,
for
sure.
and that's another layer of complexity.
Yeah, I mean, how the microbiome is getting acquired, like how much of it is through vertical transmission versus just horizontal transmission through the environment.
Yeah.
Through the diet.
Yeah.
I don't know yet. I
Yeah.
mean, that's another question that I would love to pursue, but like captive breeding programs, captivity can shift the microbiome. The diet obviously isn't natural. And so when you bring these animals out to the wild, they might have a compromise.
I know that in tortoises and iguanas, the young will eat fecal matter.
Yeah,
So that, I wonder, be interesting to see.
Yeah.
And I'm just remembering a study in which pack rats from very different deserts, but one that had creosote, one that doesn't have creosote. They just couldn't digest, the pack rats that lived in places that don't have creosote just couldn't digest
oh,
it,
wow, interesting.
just died,
Wow.
so.
Yeah, when you removed the eggs, how did you determine when to do that?
It's a good question.
Because then also, the second question is, with that, you were saying that the dissected were smaller. Is it because they were yanked a little earlier? Or-
Yeah, so the eggs weren't smaller, but the hatchlings that emerged were. So these lizard virgatus oviposits their eggs after the start of the summer monsoon rains, and they do it quite synchronously. So they'll hold their eggs at a certain point of development. The embryonic development will kind of arrest for a little bit until they get this environmental trigger, and then they lay their eggs. And so typically what I do is I hang out at the research station until we have our first rain, and I start seeing spent females in the field, and then I'll start inducing the lizards. Once the virgatus is in captivity, they won't lay naturally, so I have to give them an injection of oxytocin to get them to lay. But I'll do that, and you can palpate the lizard and feel that they're nice, turgid, ready eggs in there. And so between the start of the rains, the spent females in the field, and just kind of feeling the females and knowing that they're nice firm eggs, I acquire all the eggs of all the females on the same day, and kind of haphazard, right, which females get dissected versus induced, and so there shouldn't be any obvious bias, there. I will point out that even in the sterile environment, dissection had a reduction in just being dissected in a sterile environment, still reduced hatch
Yeah.
success. That's unlikely to be due to, missing the antifungal protection. So I think there's likely something else, happening when it goes through the cloaca. There's all sorts of cloacal glands that might be secreting things, kind of onto the eggshell surface. And so these eggs also don't have that, right? I can't say that the only thing an egg gets by going through the cloaca is a microbiome.
Yeah.
Right, there might be other protective compounds kind of getting put on. And so, yeah, but this is, yeah. So I mean, I think this difference highlights that. And I haven't been able to figure out another way to kind of go about the question.
So, if I'm remembering correctly, maybe I'm hallucinating, but wasn't the occidentalis population in the Puget Sound introduced? And if so, I wondered how it how different it was from the source population, if that was known.
If it was introduced, I was not aware of that.
Okay, it was a long time ago for me. but
Yeah.
I was thinking they were intentionally introduced, I think in Herb Brown, to do a study on their biology, because they didn't actually occur there. So he got some occidentalis from somewhere and established a population there in the Puget Sound.
Oh, in Puget Sound, direct, so not Wall,
Yeah,
Washington,
in
but in
Puget Sound,
Puget
right.
Sound.
I've heard the same thing.
I'll have to look, Adam Lachey has a paper that looks at he looked at populations of occidentalic like all over Washington and looked at the evolutionary history of them. But I would need to go back to it
Yeah.
to see.
I didn't think they naturally occurred west of the Cascades,
Yeah,
or not at that latitude.
yeah. I'll look that up.
I
Thank
think
you.
it's Herb Brown too.
Yeah.
I'm not sure. I don't want to badmouth anybody.
Yeah. All right, yeah, thanks for that. I'll look that up.
Out of curiosity, does the uric acid urate pellet have any microbial signature to it? Like, I'm gonna drag it. bunch of urics on the eggs before they go.
Yeah. We have not detected, like obvious like urea contamination of the egg. So like basically, of the swab. So like when we take a swab sample, we'll look at it and see like, is there obvious urea on it? Is there obvious fecal material on it? And then when we do our bioinformatics and look at the composition, the samples that are noted to have urea kind of mix right in with all the other samples. But the samples that have feces are, have a clear number about them. And so I haven't like, pulled apart the urea and tested it directly. So I don't know the actual answer to your question, but it seems like it's pretty similar to the cloaca if it is, because it's not, like if we need to know which samples had urea on them, they don't separate themselves out in any way from our other samples from our. All the great questions.
It's a really, really great talk. Thank you.
Thank you.
Yep.